Hi, Assalamualaikum. My name is Siti Nursha Mimi binti Cik Rahim with metric number 149055. Today I'm going to present about the assignments of industrial microbiology with the title Bioremediation using microorganisms. The key concepts of these slides are: what is bioremediation, microbes isolation, identification technique, the characteristic of the selected microbes, performance of selected microbes, suggestion, and conclusion. What is bioremediation? Bioremediation is a process used to treat contaminated media, including water, soil, and subsurface material, by decompose the pollutant by using microorganisms. The process is a clean, cost-effective, and environmentally friendly, as it is more sustainable than other alternatives. It reaches a complete degradation of pollutant and does not produce toxics as byproduct. The examples of bioremediation-related technologies are phytoremediation, mycoremediation, bioventing, bioleaching, land farming, bioreactor, composting, bioaugmentation, rhizofiltration, and biostimulation. Next is microbes isolation, the isolation and screening of strain. First, 10 grams of the contaminated soil sample was added into 200 milliliter of the mineral salt medium containing 20 grams per liter of the crude oil as the sole carbon and energy source. It was incubated in a rotary shaker at 150 rpm under sterile condition at 37 degrees Celsius for 7 days. Then 100 micrometer of the incubated culture was added to the fresh solution of the mineral salt culture containing 20 grams per liter of the crude oil. The inoculation was repeated for 3 times under the same condition. The procedure was lasted for 4 weeks, and after enrichment, 10 micrometer of the final incubated culture was spread on the nutrient agar plates containing 10 grams per liter crude oil and then incubated at 37 degrees Celsius for 24 to 48 hours. The grown single and pure colonies were cultured separately on the fresh plates and re-incubated for 24 to 48 hours. After that, the isolated colonies were picked from the plates and stored in bit extractant medium BEPM with 15% glycerol at negative 80 degrees Celsius until they were used. The identification technique were divided into two, which is biochemical test and 16S-rRNA. Under biochemical test, there are two, which are microbiome monitoring and DNA extraction and isolate confirmation by the PCR method. While under 16S-rRNA, there were sequencing method. First, microbiome monitoring. What is microbiome monitoring? Microbiome monitoring is a continuous assessment of water quality, provided that the applied analytical method is meaningful, simple, rapid, and cost-effective. For the procedure, all the isolated strains were cultured on the nutrient agar plates for 24 to 48 hours for growing pure colonies. Then the grown colonies of each strain were dissolved into 10 ml of distilled water to increase turbidity stemming from the growth of the bacteria by its optical density OD at the wavelength of 600 reach 0.5 mcfarlane standard. Next, these solutions were added to 100 ml of the sterile culture containing 2% of the crude oil and then incubated in a shaking incubator at 37 degrees Celsius for 30 days. The growth rates of all strains were assessed and compared daily by means of their level of OD 600. A separate 100 ml mineral salt culture containing 2% of the crude oil without any strain was considered as the control sample, and accordingly its OD 600 was monitored during the period. The next identification technique is DNA extraction and isolate confirmation by the PCR method. Polymerase chain reaction (PCR) is a method widely used to rapidly make millions to billions of copies of a specific DNA sample, allowing scientists to take a very small sample of DNA and amplify it to a large enough amount to study in detail, where DNA extraction was performed through the thermolysis or boiling method with some modifications. First, 400 microliter of ice-cold ethanol was added into the microtube containing nucleic acid so as the concentrate it attained from the thermolysis. Next, the solution was mildly mixed and kept in a deep freezer at -18 to -20 degrees Celsius for 10 to 30 minutes. Afterwards, it was centrifuged for 10 minutes at the maximum speed of 4000. At this stage, the supernatant was removed and the pellet was dried out fully by means of the heater block at 55 degrees Celsius. Finally, 53 microliter of sterile trace adapter buffer was poured into the microtube and the pellet was mildly dissolved. A few of the concentrated nucleic acid were used for PCR and the rest were safe frozen at -20 degrees Celsius for future applications. In order to confirm the extracted isolates that were detected by phenotypic methods, PCR assay was done using Bioremediation thermal cycle. The PCR mix contained 1 microliter of the primer, 2 microliter DNA, 12.5 microliter PCR master mix in a final 25 microliter reaction volume. DNA application was conducted in a thermal cycle under the following condition: initial denaturation at 95 degrees Celsius for 5 minutes, followed by 35 cycles of denaturation at 95 degrees Celsius for 1 minute, and aniline temperature at 57 degrees Celsius for 1 minute, and extension at 73 degrees Celsius for 1 minute, a final extension at 72 degrees Celsius for 5 minutes. Electrophoresis of the amplified DNA fragments along with 100 BP DNA ladder was carried out using 2% agarose cell. The last technique was sequencing. Sequencing in genetics and biochemistry means to determine the primary structure of an unbranched biopolymer, where one sample of each PCR product was sequenced. The data were analyzed using the Chromas software and aligned with the Cluster W program. Next is the characteristic of the selected microbes, where it could go in extreme environment polluted with high levels of petroleum combination. It also have capacity to adapt through coincidental induction of many catabolic pathway, leading to reach new ways of different pollutant bioremediation. It also able to produce catabolic enzyme effective in petroleum bioremediation. Besides that, the selected microbes also able to adapt in different types of environments like oil sludge or highly polluted soil. Last but not least, it also able to degrade crude oil. Performance of selected microbes. All 9 strains of bacteria were capable of growing in the mineral salt broth containing 2% of the crude oil as the sole source of carbon and energy. There were 3 bacteria were selected. The first one is the growth rate of strain 8, which is Pseudomonas sp, was 0.8, which is the best peaking after 23 days. Next is the growth rate of strain 5, which is Staphylococcus aureate, which shows 0.5 reading after 20 days. And the last strain is the growth rate of strain 3, which is Pseudomonas sp, show 0.4 reading after 20 days. Strains that have the highest growth rate. First, strain 8, which is Pseudomonas sp, the growth rate was 0.8. Second, strain 5, which is Staphylococcus aureate, the growth rate is 0.5. And lastly is strain 3, Pseudomonas sp, and the growth rate is 0.4. These strains belong to the gamma subclass of Proteobacteria and Firmicutes, which is able to produce catabolic enzyme effective in petroleum bioremediation. It also able to adapt in different types of environments like oil sludge or highly polluted soils. Besides that, it also have capacity to adapt through coincidental induction of many catabolic pathways, leading to reach new ways of different pollutant bioremediation. For the suggestion, there is 4 that I would like to suggest for the microbes. First, promote the vermi biofiltration for larger scales. Second, use advanced genetic engineering technology for production of larger scales. Next, enrich a culture in laboratory to isolate the bacteria with the trait of degrading crude oil. Lastly, choose a suitable microbial populations that can degrade a wide range of contaminants. The conclusions. We can conclude from the isolation and identification of new strains of crude oil degrading bacteria from Carr Island, Iran. It is shown that strain 8, which is Pseudomonas sp, has the highest reading of growth rate, which is 0.8. This strain belongs to the gamma subclass of Proteobacteria, where it able to produce catabolic enzyme effective in petroleum bioremediation. Next, it's also able to adapt in different types of environment like oil sludge or highly polluted soils. Lastly, it also have capacity to adapt through coincidental induction of many catabolic pathway, leading to reach new ways of different pollutant bioremediation. That's all from me for this presentation. Thank you.